Okay, what is glucose oxidase? Same like others previous group. The EC number is 1.1134, which is oxidoreductase. The synonym of glucose oxidase is beta D-glucose oxidase, or also known as D-glucose oxidase. The catalytic reaction is to catalyze redox reaction and catalyze sugar to produce hydrogen peroxide and gluconolactone. The cellular location is intracellular. So there are several physical properties we mentioned in here, which is the molecular mass, isoelectric point, heat stability, monomer or dimer. So glucose oxidase is found to be dimeric, and the cofactor, it contains one tightly non-covalently bound FAD cofactor monomer. Cofactor is just like the car which need fuel for it to move. So cofactor is something that help enzyme to function.
Okay, so here I would like to compare the differences and similarities of the GOX in Penicillium glaucum and Helicobacter. So roughly, you can see the differences. GOX in fungi is inhibited by pyranose oxidase, while the GOX in HAFIZAH was not inhibited by several inhibitors, but it was sensitive to HgCl2 and it precipitated in the form of mixture. So, GOX in Aspergillus niger is more sensitive to ophthalate Ag plus ion and Hg2 plus ions, while the Helicobacter, its GOX is relatively less sensitive to ophthalate Ag plus and Hg2 plus ions. So the optimal pH for fungal GOX specific in Aspergillus niger, it tends to be lower than HAFIZAH GOX. The enzyme GOX migrates faster during SDS stage for the Aspergillus niger, while in the Helicobacter it migrates slower during the SDS stage.
So as we can see, the glucose oxidase has many important applications. For example, in food processing, it acts as an additive. In food preservation, act as preservative in seafood. Then in LPS, glucose sensor or assay, and genetic engineering, like we have learned in the previous semester. So this is the addition for our knowledge is glucose oxidase can provide resistance to bacterial infection. Therefore, glucose oxidase also used to produce hydrogen peroxide for bleaching, utilizing the glucose generate, which is our detergent. Okay, here are the protocol to measure enzymatic activity for different substrates. Usually, when we say about enzymatic activity is most related to pH and temperature. So here we use the assay known as enzyme assays. GOX activity can be measured using indirect oxidation of O-deaniscidin by horseradish peroxide (HRP). So the steps we use is we prepare those three reagents, then mix the reagent, then the final reagent we take about 3 ml and add it with 0.1 ml of diluted GOX sample. So the GOX sample can be diluted if you want, if necessary, so that it within the range. Then measure measure using spectrophotometer with the adsorbent 436 nanometer. The result is the productions of D-gluconolactone and the hydrogen peroxide. The decomposition of hydrogen peroxide can be measured by using catalytic activity. To measure the effect of the enzyme activity, the pH of the enzyme is usually affected by the nature of buffer. The protocol and method to produce cell extract from Aspergillus niger. The method we use is liquid state fermentation. We use medium contain of glucose, urea, and others. First, the 5% of sample in inoculum size was added aseptically to the flask and incubate at orbital shaker enzyme obtained by filtering the ferment biomass and then subjected to centrifugation. The activity of glucose oxidase was determined at each step of replication by where an amount of 0.5 ml enzyme was added to reaction mixture. Okay, so the reason we use the liquid state fermentation because it can provide high level of glucose oxidase.
First of all, the sample with 150 ml of GOX buffer is centrifuged for 10 minutes. The protein precipitate with ammonium sulfate and then centrifuged again for 1 hour. The precipitate was suspended in 200 ml of DDH2O and concentrated to 10 ml in an Amicon ultrafiltration unit using PES ultrafiltration membrane. Next, it washed the sample with 3 times of 200 ml DDH2O. It is diluted to 50 ml with 20 molar trace buffer and then applied to super Q650 mol anion exchange resin column. And then the bound protein was eluted with 500 ml of linear gradient at flow rate 4 ml per minute. Collection of 10 ml fractions is measured and with the UV absorption 280 nanometer. High content of GOX activity were pulled and washed with 10 volume DDH2O in Amicon ultrafiltration unit. The sample lyophilized and stored at negative 20 degrees Celsius. And then dissolve the sample in minimum amount of DDH2O, and as a final purification step, the sample was applied to size exclusion column pack with cefacryl S200HR. How to ensure the protein purified is GOX? First of all, we will do the SDS polyacrylamide gel electrophoresis, and then the protein blot on nitrocellulose, and then the label, and then it labeled with specific antibody, and the process were detecting antibody, so we can get the protein of interest.
We run the agarose gel electrophoresis to separate a mixed population of DNA or proteins. It is separated by charge, size, or both. At the end, if there is a band present, means sample was contaminated. But if there is no band, means it is purified. This is the 3D structure of glucose oxidase. I find it in PDB database. Active site was determined by comparing the protein sequence of glucose oxidase extracted from different organisms. Protein sequences that has identical amino acid indicates it is important for enzyme function and usually for active site or binding site. Use the bioinformatic tools cluster IW2 to determine the identical sequence of amino acids in different organisms. This is 3D structure from Yasara. There are also other methods to find the active site. So here is what I get. A quick and dirty way to find the active site is to look for the largest cavity on the surface of the protein. Why? This is because a cavity has a relatively large contact surface which allows for many interactions such as hydrogen bonds, ionic and hydrophobic interaction. This will increase the specificity of the active site for a particular ligand compared to an active site not located in a cavity. This change in enzymatic activity can be detected by secular dichroism or CD. The function of secular dichroism is to observe secondary structure, analyzing secondary structure or conformation of macromolecules, give useful information about protein structure, the extent and rate of structural change in ligand binding. The application it can be used to study large biological molecules. GOX is large in size. Studies of protein folding and the study of chiral molecules of all types and size. Assessing the structural integrity of membrane proteins during extraction and characterization processes.